Thank you all for being here. My name is Furhad Nasserjah, and today I'll be defending my dissertation that's titled From Social Inequality to Microbial Ecology, investigating how socioeconomic status and medical treatment shape the human gut microbiome. Thank you to everyone for being here, and thank you to everyone on the committee, Dr. Koizumi, Dr. Andalibi, Dr. Chen, Dr. Equina. Thank you all for being a part of such an experience for me, so I really appreciate it. So starting off, here's the abstract for the dissertation. And so health outcomes often depend on social and economic inequality. And so people's access to care, their ability to prevent disease, and even their biological well-being are tied to those factors. And the gut microbiome, which is the bacteria living inside of us, is a great way to study that link. This project combines social science methods with clinical and ecological tools to explore how economic standing and insurance type affect gut microbial diversity. And so I'll do this in 3 main chapters that we'll go through together, and they're going to build one system showing how inequality produces measurable biological change in human microbiomes. And so here is the causal diagram behind my study, and I start with socioeconomic status, which is SES, because SES is really the upstream factor that shapes almost everything else. And SES affects which type of insurance people have. Insurance then affects treatment access and quality, and that's what eventually shows up in the microbiome. So that's the core chain that I'm trying to study. And SES also influences a set of confounders and that I needed to account for. And so people with lower SES often have different diets and higher stress, more comorbidities, and more antibiotic exposure. And all 3 of those factors have a direct effect on the microbiome. Comorbidities also affect treatment access, and all of this can bias the relationship between insurance and microbiome outcomes if I don't control for them. And so I also include this direct SES to microbiome arrow because SES shapes the microbiome through environmental exposures, stress, and diet, even outside of the healthcare system. So overall, it's just showing the assumptions behind my identification strategy, and it makes it clear what needed to be controlled for, where bias can enter the model, and why isolating the effect of insurance requires adjusting for SES and these upstream factors. And so the motivation and importance of this project was that SES is one of the strongest predictors for health outcomes and influencing disease risk, mortality, access to healthcare. However, the biological mechanism behind those effects isn't fully understood. In addition, the gut microbiome plays a central role in metabolism and immune function and inflammation, and it's highly responsive to environmental exposures like diet, stress, and healthcare access. And so because of this, it provides a strong candidate for understanding how social inequality translates into biological differences. And so here is the overview of the 3 chapters, and Chapter 1 is establishing overall microbiome patterns across studies using a systematic review and meta-analysis. Chapter 2 evaluates those patterns in patient-level data, and Chapter 3 uses the same patient cohort in Chapter 2, and it examines how microbial differences appear on an ecological networks level. And so starting off with Chapter 1, the goal is to determine whether there is a consistent relationship between SES and the microbiome across prior studies. And so this chapter focuses on 3 levels of microbiome analysis. It has to do with alpha diversity, which reflects within sample diversity, beta diversity, which captures differences in community composition between groups, and taxonomic composition, which examines the relative abundance of specific microbial taxa. And so the primary research question for this chapter is, does SES influence gut microbiome diversity and composition in US populations? And so doing a little bit of background research, it suggests that SES is associated with microbiome differences, although some of the results did vary. For alpha diversity, some studies showed that lower diversity in lower SES populations, while others showed mixed results depending on what diet and population there was. The beta diversity findings are a little bit more consistent, and they showed that microbial communities did differ across socioeconomic groups. And at the taxonomic level, studies often reported changes in the taxa, with things like increasing in Prevotella species, decreases in short-chain fatty acid producing bacteria, in lower SES populations, which those are all important for gut health and metabolic function. And so I conducted a systematic review and meta-analysis of US-based peer-reviewed studies. And so this chapter is following the PRISMA 2020 guidelines for study identification, screening, eligibility assessment, and synthesis. And so my search included PubMed, Google Scholar, and 2 different LLM-assisted discovery tools, which were ChatGPT and Gemini, as supplementary search tools. And so the overall design is combining a quantitative meta-analysis for alpha diversity with a structured narrative synthesis for beta diversity and taxonomic abundance. And that design was necessary because of the heterogeneity in populations, the SES definitions, and the microbiome measurements that were the methods that they used across those different studies. And so here is the PRISMA flow diagram. And so this figure is showing the PRISMA flow that summarizes the full screening process from the initial search results down to the final set of included studies. And so using all of the different databases and LLMs, there was a total of 765 relevant articles, which then went down to 8 studies for the inclusion criteria for overall narrative synthesis. And then of those 8, 4 of them were eligible for quantitative meta-analysis. And so this is also a good place to briefly mention the role of AI in this chapter. AI tools were used to assist with the study identification, the screening, and eligibility assessment. However, all the outputs I manually verified to ensure the accuracy. And so it was used as a support tool rather than a replacement for human evaluation. And it was also used for minor grammar checks in the writing process. And so this table here is summarizing the 8 included studies for the narrative synthesis. And so when interpreting this table, it was just here it was important to note the variability that was across the studies, which explains the design choices for the chapter to split it up into a meta-analysis versus narrative synthesis. And so for data analysis, alpha diversity was analyzed using Hedges G, which standardizes differences across studies, and a random effects model was used to account for variability across the populations. Effect sizes were calculated using Hedges G to account for the small sample bias, and they were pooled using inverse variance weighting under a Dare-Siminoid random effects model to account for between study heterogeneity. And so to maintain independence and avoid inflating precision, a 1 effects per study rule was applied, meaning that each study contributed to only a single effect size to a meta-analysis. And so alpha diversity was selected for quantitative meta-analysis because it did produce continuous and comparable outcomes across the studies, whereas beta diversity was assessed using distance metrics like Bray-Curtis and Unifrac with Permanova to test for statistically significant differences in the community composition. So this table is now showing the subset of the studies included in the meta-analysis only. And so again, this 1 effect per study rule was applied to maintain independence and to avoid inflating precision so that no single study disproportionately influenced the results. And so using this approach, I then evaluated the pooled results starting with alpha diversity. And so this is a forest plot, and it's showing the results for that alpha diversity, including individual study effect sizes and the pooled estimates. And so each line is representing a study where the dot shows the effect size and the horizontal line represents the 95% confidence interval. And so when interpreting this figure, I looked at both the direction of each effect and whether the confidence interval crosses 0. And so this is to evaluate both the consistency of the findings across the studies and the overall association between SES and alpha diversity. And the pooled estimate appeared on the positive side, but I'm going to walk through the magnitude and statistical significance of those results on this very next slide. And so looking at the pooled results, the effect size was 0.219, which indicated a small but statistically significant association between higher SES and higher alpha diversity. And so the confidence interval didn't also cross 0, confirming that statistical significance. The I-squared value here represents the proportion of variability across studies that reflects true differences in effect sizes rather than random variation. And that was at 81.9%, which indicates substantial heterogeneity, meaning that the effect of SES on microbiome diversity varies across studies, likely due to the differences in the studies' different populations, their SES definitions, and analytical methods. So while the overall association was positive, that strength varies across different studies. Now, moving on to summarizing the beta diversity results across the studies, the beta diversity was assessed using distance metrics, such as Bray-Curtis and Unifrac, and statistical significance was tested using Permanova. And so when interpreting this table, I tried to focus on the Permanova results and how consistent those findings were across the studies. And so all of the studies except for one of them showed statistically significant differences in microbial community composition across the SES groups, indicating that SES is associated with shifts in the overall community structure. And finally, for the results for Chapter 1, here is a summary of the taxon level associations across the studies. When interpreting this table, I focused on identifying patterns that appeared across studies rather than an individual statistically significant finding. Across the studies, there were recurring associations involving important metabolic taxa like Prevotella, bacteroids, and SCFA producing bacteria, which had to do with metabolic functions. And so in several of the studies, lower SES is associated with higher Prevotella abundance and lower levels of SCFA producing bacteria, even though those patterns are not consistent across all of the studies. So this slide kind of shows that SES may be linked to shifts in microbial composition and function. And so with the chapter analysis, there are some limitations to consider. First, there were high heterogeneity across the studies. Second, the number of studies included was relatively small. It still matched the criteria for a PRISMA 2020 guideline for the number of quantitative studies that were in the meta-analysis, but it was still relatively small. And third, there were methodological differences across the studies. So overall, this chapter showed that the quantitative meta-analysis demonstrated a small but statistically significant association between higher SES and higher alpha diversity, differences in microbial community composition across the SES groups, and there were some reoccurring patterns in the microbial composition involving the metabolically important taxa. And so these findings support the idea that SES influences microbiome through environmental, behavioral, and structural pathways. And so on to Chapter 2, which builds directly on the findings from Chapter 1. So while Chapter 1 established broad patterns across prior studies, those results were based on aggregated data from different populations. And so the goal of Chapter 2 is to determine whether those same relationships between SES and the microbiome are observed in patient-level data. Therefore, the research question for this chapter is, does SES influence gut microbiome diversity and composition in patient-level data? And so, as shown in Chapter 1, there was some strong evidence that SES is associated with differences in the microbiome. Studies used in our syntheses in Chapter 1 showed associations between SES and alpha diversity. Again, although some of those results can vary depending on the population. Other studies demonstrated differences in microbial community structure across socioeconomic groups. And at the taxonomic level, some studies show shifts in key metabolic bacterial groups. However, these findings were largely based on population-level studies as opposed to patient-level data sets. And so this chapter uses de-identified stool microbiome data from Virginia Bioanalytics kidney transplant cohort. And so the full data set included 27 total patients, with 22 of them having paired pre- and post-transplant samples, which allowed for within-patient comparisons over time. And so the main exposure for this chapter is insurance type, specifically whether it was Medicaid versus non-Medicaid insurance. And so to the right is table 2.6.1, and on the side, it's pretty much showing that it's a visual of the cohort data, some of the cohort data, and the descriptive analyses that were done to assess a baseline comparability between insurance groups, and this helps contextualize the results and identify any potential confounding. The outcomes included alpha diversity metrics to capture differences, different aspects of within sample diversity, beta diversity, similar to the last chapter, evaluated using Bray-Curtis dissimilarity and PCOA to assess differences in the community composition between samples, and then exploratory genus level taxonomic analyses to examine changes in specific microbial groups. So for change in alpha diversity by insurance group, I was using Mann-Whitney U tests with effect sizes because the data were not normally distributed and the sample size was relatively small. And for beta diversity, I used PermANOVA for significance and PermDisp, which is a statistical test that assesses whether groups differ in variability to ensure that any differences detected were not driven by unequal dispersion between groups. So starting off is with the alpha diversity. And so the left side going diagonally down is figure 2.6.1, and that's showing paired pre and post-transplant alpha diversity. And then figure 2.6.2, which is beside it, again, horizontally moving down, that's showing alpha diversity changes after transplantation was stratified by insurance status. So while interpreting these figures, it's best to focus on whether diversity changes consistently over time and whether those changes differ between Medicaid and non-Medicaid patients. So there was a statistically significant decrease in Shannon diversity across the transplant time points. However, this was not consistent across the other diversity metrics for alpha, and it didn't differ by insurance status. With the insurance stratified analysis, the effect size estimates were imprecise, and the confidence intervals were pretty wide, and that suggests that the cohort was underpowered to detect some level of modest insurance-associated differences. So main takeaway from this slide is that alpha diversity didn't show robust evidence for insurance-related differences in this cohort. Moving on to the beta diversity, this figure is showing beta diversity by insurance status using Bray-Curtis dissimilarity and a PCOA plot. And so each point is representing a sample, and the distance between the points reflects differences in overall microbial community composition. So visually, I'm looking for clustering or separation between Medicaid and non-Medicaid groups. And here, the two groups show substantial overlap with no clear separation, which suggests that insurance data didn't prove a distinct community level pattern in this cohort. The broader pattern here is higher inter-individual variability rather than a strong group separation. Continuing on with that, this is showing the PermANOVA results for the community composition. And so for both transplant time point and for insurance status, there was no statistically significant difference when looking at the p-value or the F statistic, which reflects the ratio of between group or within group variation, or the really small R-squared value, which represents the proportion of variance explained. I also assessed homogeneity of dispersion using PermDisp, which was, when you look at the F statistic and the p-value, the dispersion was not significantly different between pre- and post-transplant samples or between the insurance groups. So overall, these results are indicating that neither transplant time point or insurance status was significantly associated with community level variation in this cohort, and that the lack of significance was not due to unequal group dispersion as shown by the PermDisp results. And finally, for the results of Chapter 2, this slide is summarizing the taxonomic results at the genus level. So the analysis was conducted to evaluate whether specific bacterial taxa differed across transplant status or insurance groups. So when interpreting these results, the focus is on whether any taxa show consistent and statistically significant differences across the groups. And so in this cohort, some nominal unadjusted differences in the genus level abundance were observed. However, none of these differences remain statistically significant after the FDR correction, which that's adjusting for multiple comparisons to reduce the likelihood of a false positive. And so in addition, there was no consistent pattern linking specific taxa to insurance status across the patients. And this suggests that while there may be subtle shifts in microbial composition at the taxonomic level, these findings are exploratory and not statistically robust in this data set. So in this chapter, there were some limitations to consider. First, the paired samples were small, which limited the statistical power. The second thing is that the insurance group, there was an imbalance with relatively few Medicaid patients, which made it difficult to detect modest differences. And third, the cohort showed substantial inter-individual variability, especially in community composition. And finally, because this was a kidney transplant cohort in a very specific clinical setting, the findings may not fully generalize to a broader population. So overall, the results from this chapter didn't provide strong evidence that insurance status was associated with the differences in gut microbiome after the corrections, and it also didn't have significance with the community composition. So the main conclusion from Chapter 2 is that this cohort showed high variability and limited power, and that larger studies might be needed to determine whether insurance-associated signals can be detected more clearly. And so Chapter 3 is building on the previous chapters by moving beyond diversity and composition to examine the microbiome as an ecological system. So in chapters 1 and 2, I focused on which bacteria are present and how they differ across the socioeconomic groups. Chapter 1 compared aggregated and varying population data from different studies, and then Chapter 2 compared a specific patient cohort data. However, those chapters' analyses don't capture how bacteria interact with each other. And so in this chapter, I'm using the data from cohort 2, the data from Chapter 2, from the cohort in Chapter 2, and I'm modeling the microbiome as a network where nodes represent taxa and edges represent associations between them. And so this allows us to evaluate not just composition and diversity, but the structure and the organization of the microbial ecosystem, which is valuable because interactions between microbial taxa are important for the overall microbiome because bacteria influence each other's metabolisms, their stability, and their overall ecosystem behavior. Thus, the research question for this chapter is, does socioeconomic context and medical treatment influence microbiome network structure? So the microbiome is not just a collection of individual species, but it's an ecological system where interactions between taxa determine stability and resilience. And so some of these studies show that higher connectivity is often associated with greater stability and resilience in microbial networks, depending on the type of interaction that's present. Other studies showed that fragmented networks in these microbial communities with fewer connections are more vulnerable to stresses. Stresses like the ones our kidney transplant cohort from chapter 2, which we're using here again, would be undergoing. And so most microbiome research has focused on diversity and composition, but few studies have examined ecological network properties. And there is especially limited work connecting socioeconomic factors to microbiome network structure, which is what this chapter is addressing. And so this chapter uses microbiome data again from the same kidney transplant cohort as chapter 2. The data consists of genus level relative abundance profiles. And before conducting the network analysis, I filtered the data to reduce sparsity and to focus on more consistently observed taxa. CLR transformation was then applied to account for the compositional nature of the microbiome data, where relative abundances are constrained and interdependent. And to study the analysis, I organized the chapter into 4 different stages. With alpha and beta diversity for stage 1, chapter 2 evaluated between group differences by insurance status, whereas chapter 3 is focused on diversity on within cohort temporal changes, and it extends that analysis to ecological structure. And it captures patterns that hopefully wouldn't be detected by just diversity metrics alone. And then stage 2 involves network construction using Speakeasy, which estimates conditional associations between taxa while accounting for indirect relationships. Using the STARS approach, stable network structure was identified, and selecting a level of sparsity that minimizes variability across the subsample networks. With stage 3, it evaluates network topology, including metrics like edge count and density and modularity. And it includes validation using SparseCC as a sensitivity analysis, and to estimate correlations between taxa while accounting for the compositional constraints. And lastly, stage 4, that's comparing the generated transplant networks from our patient cohort to a healthy reference data set from the Human Microbiome Project. So using this framework, we're going to start off with stage 1, starting off with alpha diversity. And so when interpreting this figure, I focus on differences in central tendency and paired changes across the individuals. And so Shannon diversity again showed a statistically significant decrease following transplant, although the magnitude of those changes are modest and there is substantial variability across those patterns. Whereas the Cal1 richness and the Simpson diversity also trends downward, but their changes weren't statistically significant. And so overall, this is indicating that transplant may be associated with a modest reduction in microbial diversity at the group level, but these changes aren't consistent across all diversity metrics, and they do vary between individuals. This figure is showing the beta diversity using the PCOA plot, and again, each point represents a sample, and the distances reflect differences in the microbial composition. And so when interpreting this, I'm looking for clustering or separation between the pre and post-transplant group. Visually, the separation is pretty modest, with a lot of overlap between the samples. However, the Permanova results showed a statistically significant difference with a small p-value and R-squared value. And so that indicates that transplant status does explain a small proportion of variation in the microbial composition. This slide is showing a figure that shows the relative top 15 most abundant genre in pre- and post-transplant samples. And so when interpreting this figure, I'm looking for shifts in dominant taxa between conditions. And so what was interesting was there were changes in relative abundance of several of those metabolically key genera following transplant, like the Prevotella, the bacterioids, and any of the SCFA producing bacteria. However, these changes are variable across the taxa and across some of the samples, and so they don't show a consistent directional pattern. Moving on to stage 2 is network construction, and so these figures are showing the generated microbial ecological networks before and after transplant using Speakeasy. And so each node is representing a genus, and the edges represent conditional associations that are inferred from the data. So we can observe differences in how taxa are organized and clustered between the two networks. And so visually the post-transplant network appears to be less connected and more compartmentalized, with tighter clusters and fewer connections between the groups. And the key conclusion about changes in connectivity and fragmentation really come from the quantitative network topology metrics, which I'm going to show in all of the following slides, which is really where stage 3 of this chapter begins. And so moving to stage 3, this table has those network metrics and shows the top microbial hub taxa based on network degree. So when interpreting this table, I'm focusing on which taxa have the highest number of connections within the network. And so these hub taxa represent highly connected nodes, meaning that they're centrally positioned within the inferred ecological network. I also observed changes in which taxa function as hubs between pre- and post-transplant networks, suggesting that shifts in which taxa occupy central positions in the network. So these changes are consistent with the broader pattern of reduced connectivity and altered ecological study following the transplantation that was in the visual representation. And this table is quantifying the differences between pre- and post-transplant networks using Speakeasy. So when interpreting this, I'm trying to focus on the key metrics such as edges, the count, the edge count, the density, the average degree. And so when we look at those, the number of edges decreases after transplant, which indicates fewer inferred associations between the microbial taxa. The average degree and network density also decrease, reflecting reduced overall connectivity. Modularity increased, which indicates greater separation in the subnetworks. And these patterns are consistent with a network that is less connected and more fragmented following transplant, which is what we're expecting. This slide here is showing the results from the sparse-CC network analysis that was used for validation. And so when interpreting this, I'm looking for whether the same patterns are observed with Speakeasy and if they're replicated. So the sparse-CC results fortunately showed that the same overall pattern, edge count, average degree, and density decreasing, showed that same trend, and modularity increasing, which again was the same trend. So these results are consistent with the patterns observed using Speakeasy, suggesting that the observed network fragmentation is not dependent on a single inference method. Onto the final stage of the results, stage 4. This slide is comparing our generated transplant networks to a healthy reference network. So when interpreting this, I'm comparing connectivity, clustering, and modularity. And the healthy network is showing higher clustering and a more integrated structure. Whereas in contrast, both the pre- and post-transplant networks of the highly stressed individuals in the kidney transplant cohort, they showed reduced connectivity, lower clustering, and higher modularity. And so another thing was that the post-transplant network shows the greatest degree of fragmentation. And so all of these patterns suggest that transplant-associated microbiomes are less integrated and they exhibit altered ecological organization compared to a healthy microbiome. And so onto the key limitations of this chapter. First, network inference reflects statistical associations between taxa rather than confirmed biological interactions. Second, the cohort sample size was still, again, relatively small, which again limits statistical power and may reduce the ability to detect more subtle or consistent patterns in some of those tests. And finally, the study design includes both cross-sectional and paired elements, which introduces constraints in how changes can be interpreted across the individuals over time. And so the summary of the key findings in Chapter 3 is that at the diversity level, Shannon diversity showed a statistical significant decrease following transplant. At the community level, microbial composition shifts significantly. At the network level, there was a observed consistent reduction in connectivity and an increase in modularity and fragmentation. And so these all suggest that after transplant, the microbiome was less coordinated, it's more disrupted, it's likely reflecting the effects of the immunosuppression, the antibiotics, the physiological stress, which can weaken normal microbial interactions and lead to a more unstable ecosystem. And so importantly, these ecological changes would have not been fully captured had we only used the diversity metrics that were analyzed in Chapter 1 and 2. And so here are the summaries of the findings across all three chapters. In Chapter 1, found that SES is associated with the microbiome at the population level, including a statistically significant relationship with alpha diversity and consistent differences in community composition across the studies. In Chapter 2, these effects were strongly detected in a patient-level transplant cohort. They were not strongly detected. And these results were variable and not statistically significant, which likely reflected the high inter-individual variability and that limited sample size. And in Chapter 3, I found that transplants and associated treatments were associated to changes in the microbial network structure. And so in conclusion, this work was trying to show that social inequality is associated with variation in the gut microbiome, but these effects depend on the level of analysis, where with clearer signals at population and ecological levels than usually at the individual level. And so these findings suggest several directions for future research. First, the value of larger, well-powered longitudinal studies that are needed to better understand how socioeconomic and clinical factors influence the microbiome at the individual level. And second, future studies should move beyond diversity alone and incorporate network-based approaches to better understand microbiome organization and function. The results point toward the need for integrated multi-level analyses that combine things like population level, individual level, and ecological network perspectives. So I would like to sincerely thank each and every one of you, Dr. Koizumi, Dr. Andalibi, Dr. Chen, Dr. Ekwana, everyone else that's here, Mehdi, Monty. Thank you all for being here. It's been an honor for me to work with you all, and I'm extremely grateful for this opportunity and experience. So that's the end of the slides.
Great presentation. Thank you, Fahad.